Hello, my name is Aschenbrenner, and I'm a postdoctoral scientist at the Nuffield Department of Experimental Medicine and the Translational Gastroenterology Unit of the University of Oxford. And it's my pleasure today to introduce our study that was recently published in the journal Gut, and it has the title Deconvolution of Monocyte Responses in Inflammatory Bowel Disease, reveals an interleukin-1 cytokine network that regulates IL-23 in genetic and acquired interleukin-10 resistance. So in this study, we set out to model interleukin-10 dependent regulation of inflammatory immune cell activation and interleukin-23 synthesis in patients with IBD and healthy individuals. For that, we stimulated peripheral blood mononuclear cells with pattern recognition receptor ligands such as MDP or LPS, or activated T cells with anti-CD3 and anti-CD28 beats in the presence or absence of interleukin-10 signaling blocking antibodies. And we then used different readouts, including screening of secreted factors in cell culture supernatants, transcriptomics, single-cell RNA sequencing, and flow cytometry to investigate the respective cellular responses, where our main aims were, first of all, to identify cellular sources and transcription signatures of IL-23, to define key regulators of IL-23 synthesis, and to translate the experimental and mechanistic insights from our study into innovative patient stratification strategies for personalized medicine. So by screening PBMC cell culture supernatants, we first established a signature of IL-10 regulated proteins in the context of LPS stimulation. And this signature was really led by IL-23 as the top differentially regulated protein. We next used flow cytometry to show that monocytes were the major source of IL-23 and interleukin-10 in these cultures under condition of LPS stimulation and interleukin-10 signaling blockade. When we looked at the flow cytometry data in more detail, we could see that IL-10 and IL-23 were actually produced by distinct monocyte subsets upon LPS stimulation and interleukin-10 signaling blockade. So to gain further insight into the heterogeneity of the monocyte response under conditions of LPS stimulation and loss of IL-10 signaling, we applied single-cell RNA sequencing, and this revealed 8 clusters of monocytes across 3 conditions, out of which cluster 4 was particularly interesting to us since it specifically contained IL-23 expressing monocytes, and interestingly, these monocytes co-expressed the interleukin-1 receptor 1. Other markers such as IL-10, IL-10RA, IL-10RB, or IL-1 were not expressed in a cluster-specific manner, but expressed across clusters. So to then translate IL-10 regulated signatures of interleukin-23 and IL-10 expressing inflammatory monocytes to intestinal IL-23 transcription and to deconvolute IL-23 expression in intestines, Stephen Sansom applied a weighted gene correlation network analysis to tissue transcriptome data from intestinal biopsies of juvenile onset Crohn's disease patients and controls of the risk cohort. And this analysis revealed 2 conditions of intestinal IL-23 expression. One, where IL-23 was expressed alongside tumor necrosis factor in healthy and non-inflamed tissue samples, and where IL-23 expression correlated with signatures of lymphocyte differentiation. The second condition of IL-23 expression was identified in active Crohn's disease patients with macroscopic inflammation and deep ulceration. And in this case, IL-23 was expressed alongside IL-1, IL-6, and TNF, and correlated with signatures of inflammation and inflammatory cytokine production and immune cell differentiation. So since in a risk cohort, no genetic defects in the IL-10 signaling pathway had been identified, we hypothesized that in patients with macroscopic inflammation, deep ulceration, and intestinal epithelial barrier breakdown, monocytes would become directly exposed to intestinal microbes, and that in turn could affect their interleukin-10 response. So to test this hypothesis, we stimulated peripheral blood monocytes with either LPS or heat-killed bacteria and analyzed IL-10 responsiveness in terms of STAT3 phosphorylation. And indeed, monocytes that were exposed to whole killed bacteria compared to LPS stimulation were less responsive to interleukin-10 stimulation, as indicated by decreased STAT3 phosphorylation. And these cells produced interleukin-23, suggestive of an acquired interleukin-10 resistance. So to gain further insights into the regulators of IL-23 synthesis in human monocytes, we next investigated the co-regulated cytokine network and performed perturbation experiments. And through this approach, we could confirm interferon gamma and IL-12 signaling as positive regulators of monocyte interleukin-23 synthesis. And we could identify interleukin-1 signaling as an essential factor for monocyte IL-23 production. Furthermore, we could show that interleukin-1 signaling blockade is really specific for regulating IL-23 producing monocytes at the population level. And in collaboration with Jonica Janssen, we could establish a model of interleukin-23 regulation by a network of monocyte cytokines. And this network analysis revealed interleukin-10 as a negative regulator of IL-23 and IL-1, in particular interleukin-1 beta, as a positive regulator of interleukin-23 synthesis in human monocytes. So in summary, our study revealed the presence of an interleukin-10 regulated inflammatory monocyte signature in a subgroup of patients with Crohn's disease and macroscopic inflammation and deep ulceration. Patients with this transcriptional signature are less likely to respond to anti-TNF therapy. And interestingly, in these patients, IL-23 may be produced as a consequence of direct monocyte exposure to intestinal microbes, thereby inducing an acquired state of interleukin-10 non-response. Taken together, our results suggest that this subgroup of patients with inflammatory bowel disease would most likely benefit from therapeutic strategies that directly target interleukin-23 or that alternatively target interleukin-1 upstream of monocyte IL-23 synthesis. So with that, I would like to thank all our colleagues and collaborators and funding bodies who have contributed to this study. I would also like to thank all the healthy volunteers, patients, and their families for contributing to this study and for making this research possible. And thank you all for your attention. We very much hope that you will enjoy reading our paper.